Hello and welcome to Chapter 10 with Miss Hook. The purpose of this chapter is to look over the different types of muscle. We'll then specifically focus on skeletal muscle, looking at its anatomy and physiology. Okay, so reminder, 3 types of muscle tissue: skeletal, cardiac, and smooth. Skeletal is found attached to our long bones. Each cell goes from one side of the muscle to the other. They're very long and they all run the same direction. They've got multiple nuclei here from cells that fuse together during development and lots of striations. Smooth muscle, these individual cells have a thickened middle with a very large nucleus, no striations visible. And cardiac muscle cell, striations, intercalated discs, and they branch. Okay, so here we can see the microscopic view of them from histology. Okay, and then I would have you fill out this table. You can pause the video and fill it out on your own. And once you finish that, you can go on to the next slide. And a reminder, by control, I mean is it voluntary or involuntary? Nice animation. Okay, special characteristics of muscle tissue. First, it's excitable. So this means it can receive and respond to stimuli. So like when your nervous system sends the signal down to your muscle cells, they get that signal, they spread it across their membrane, ions flow in, they contract. So the second one is contracting. What does that mean? It means the cells get smaller. They cause movement. Last 2 characteristics, extensibility and elasticity. Extensible, they can stretch. Elasticity, after they stretch, they recoil back to their original size. So like Play-Doh is extensible, you can stretch it, but when you let it go, it's not going to go back. So it's extensible but not elastic. But a rubber band, you can stretch it, and then when you let go of that rubber band, it recoils. So it's elastic. Functions of muscles, movements of bones or fluid. So we're able to move our whole body. We're able to move blood through our blood vessels by contracting or relaxing them. We're able to move air through our lungs through different areas by contracting our bronchioles. We're able to move our digestive food wastes throughout our body. Second, they help us maintain posture, body position, allow us to do communication through our facial expressions and arm gestures. 3, they stabilize joints. So for example, the shoulder joint, we give up some of that stability to allow larger rotational function of our shoulder. So we have the rotator cuff muscles that go around there to help keep it in place, so it's less likely to be dislocated. And then 4th, heat generation. Every time we contract our muscles, we use ATP. We break one of those high energy phosphate bonds, and it causes movement. But a lot of that energy is released as heat, about half of it. So each time we contract, we're also generating heat. And we can use that to stay warm. So like when you're cold, you start shivering so you can warm up. Okay, 3 questions for you to go through to make sure you understand what we've covered so far. Now let's move on to more about skeletal muscle specifically. Okay, so skeletal muscle components. You can see we've got a bone and the muscle attached, so we're looking at skeletal muscle. And remember, tendons connect muscle to bone. And there are different levels of organization within each muscle. There's the individual cell, a group of cells, and then the whole muscle. And around each you've got a layer of connective tissue. So around the whole outside, you've got the epimysium. Epi, above, goes around. And then around each bundle or fascicle, you've got the perimysium. So when you eat roast beef and it's all nice and stringy, each of those strings is a fascicle, a group of cells. And then you've got the individual cell. So you've got the cell membrane there, and right on the outside of it, you've got the endomysium. Endomysium surrounds the cell. These are all layers of connective tissue, and they come together to help form those tendons, or we'll look at aponeuroses, these flat sheets of connective tissue. Okay, and looking a little bit more at how they're formed, we talked about they've got multiple nuclei per developed cell. They start out from myoblasts, and then you've got those cells joining, fusing together to form one really long cell, which is mostly going to be made up of these contractile proteins, but pushed to the side, you've got what are called peripheral nuclei. Okay, now we're going to look at the microscopic anatomy of a muscle. Skeletal muscle cells have their own special name for pretty much every part of the cell. In fact, skeletal muscle cells are generally called skeletal muscle fibers. And around them, instead of the plasma membrane, you've got the sarcolemma. And then instead of endoplasmic reticulum, you've got the sarcoplasmic reticulum. That's going to be really important for contractions and holding calcium. And then you've got a myofibril. Myofibrils make up the cell. That's where the contractile proteins are located. And within the myofibril, there's this repeating pattern of the proteins. And each part of the protein is called a sarcomere. And we'll look at the parts of that as well. Skeletal muscle cells also have lots of mitochondria, so they can make lots of ATP. At any one time, they've only got about 2 seconds worth of ATP stored, so they have to be ready to make more. So going back to macroscopic, here's a really nice view of the different layers of organization of connective tissue. This should look a lot like meat that you're preparing from animals to eat. So on the inside, endomysium, single cell, paramysium, surrounding the fascicles or bundles of cells, and then epimysium, surrounding the whole muscle. And then all three layers come together to form a tendon, or an aponeurosis, like a large sheet that you've got of tissue. Okay, and microscopic anatomy, we're going to look at how the cell membrane is called the sarcolemma. You've got sarcoplasmic reticulum, these myofibrils with contractile proteins, mitochondria, myoglobin, which is like hemoglobin, but in our muscles that holds oxygen. So the mitochondria can use it to make more ATP by taking the oxygen and glycogen, that sugar, and breaking it down. Okay. So skeletal muscle cells are also called skeletal muscle fibers. They're these long cells that go the whole length of the muscle. Lots of nuclei, lots of mitochondria. Plasma membrane, sarcolemma. Plasma, lemma. Sarco has to do with muscle. And so the cytoplasm is sarcoplasm. And then your sarcolemma, your plasma membrane, it has these deep invaginations of tubes that go into the cell. They're called T-tubules. This is so that when we get an electrical signal into the muscle cell, it can spread through the whole cell as fast as possible so that you've got all the proteins in the muscle contracting at once, shortening at once, so we've got the most efficient contraction possible instead of one portion shortening and then the other. Okay. So here you can see those sarcoplasmic reticulum right next to the T-tubules going in. Okay, so myofibrils, those are the long rods that make up most of the cell. And they are made up of myofilaments. So those are the contractile proteins. The thick myofilament, the thick contractile protein, is myosin. The thin filament is actin. And what happens is myosin binds to actin, they pull each other together, and the cell shortens. Okay, so each of these rods is a myofibril, and it's made up of the thin and thick filament, the actin and myosin. Okay, so this shows you what they each look like. Myosin is the thicker one, the purple. That's got these like golf club heads on them. And actin is this thin yellow band. What happens is the myosin head binds to the actin and pulls it. It pulls actin from both sides, bringing the sides closer together so they shorten. Normally, myosin can't bind to actin. There's this block. You can see the troponin there. And what happens is when you get the electrical signal from the nervous system, calcium comes in, Ca2+. And what that does is it removes the block. So now myosin can bind to actin, use ATP, and contract the muscle. When you're done contracting, the sarcoplasmic reticulum takes the calcium back up and holds on to it. There's a couple questions for you there. Review what we just covered, and then move on when you're ready. Now we're going to talk about sarcomeres. Actin and myosin do this repeating pattern, and each section of the repeating pattern is called a sarcomere. It's got all the parts of the myofibril. It's like the functional unit there. So this is one sarcomere from blue line to blue line. So each edge is called a Z-disc. I remember Z is at the end of the alphabet. It's on the end of the sarcomere. And that's a stabilizing protein that connects to actin, the thin filament. The middle line is called the M line. M line is in the middle. That's what the myosin is bound to. Myosin is the dark red here. You can see M line, myosin, Z line, actin in yellow. And then the whole portion that has myosin is called the A band. The portion without is called the I band. Myosin is really thick, so the A band is going to look darker. The I band doesn't have any myosin, less thick protein. It's going to look lighter. So I like to think of A is kind of a thicker letter than I. I is really thin. So that's where you've only got thin filament. It looks lighter. And the area of overlap where you have both is the darkest. That's part of the A band. Okay, so organization, let's remind you, you've got the whole cell, the fiber, made up of those rods called myofibrils. And on them is a repeating pattern, the sarcomere, made up of myofilaments. Okay, so here you can see a real microscopic view of the sarcomere. The Z line attaching the actin, the M line attaching the myosin. So this portion is the dark portion, the A band. There's the I band, the light portion. Okay, so then what happens when the muscle contracts is the myosin heads bind to the actin on each side, and they start pulling the actin more into the middle. So these Z-discs move inwards, and that shortens the muscle. The Z-discs get closer together, there's more overlap of the myosin, they contract. So like looking back at this picture again, you could see how the heads could bind, pull the actin in from each side. With my students, I like to have them have two people be actin, one person in the middle be myosin, and have them pull them closer together. Okay. Review those questions and move on. Okay. So muscle proteins. We've talked about myosin and actin. Troponin and tropomyosin are the regulatory ones. Those are the ones that make the block, so that unless calcium is present, our muscles aren't contracting. We can stop the contraction. And then there are multiple structural proteins that make up the Z line, M line. The myosin pulling on the actin, bringing the Z discs closer together that we were describing, is called the sliding filament mechanism. And let's look at that more. So here we've got relaxed muscle, very little overlap, partially contracted, slight overlap, and then fully contracted, full overlap. So if you look at those muscles under the microscope, you can see the Z-disc to Z-disc space is the biggest on the first one when they're relaxed, and each time those Z-discs get closer together, the sarcomere shortens. And that I band, where there's only actin, gets shorter and shorter. And you see the same thing happening with the H zone, where there's only myosin that gets smaller and smaller.